Okay, everyone, here we are for the second part two video lecture of chapter 5 on macromolecules. You'll recall at the end of the first part of this, we had talked about, wrapped up talking about carbohydrates made up of monosaccharides, which are the monomers. You link monosaccharides together, you get polymers called polysaccharides. The bond between them is called a glycosidic linkage. We talked about all that stuff. Next thing we're going to talk about are a category of macromolecules called lipids. Now, lipids are also referred to as, or known more generally, as fats and oils. Okay, fats and oils. Now, I will say at the outset here, they are the one category of large biological molecules that are not built on that monomer-polymer plan we talked about with polysaccharides, for example. So we're getting away from the monomer-polymer plan for a little bit. We're going to talk about something different, the way that lipids and fats and oils are built. Then when we get into proteins and nucleic acids, as the second half of this chapter, we'll talk again about monomer-polymer because that's how those molecules are built. Lipids are not built that way, all right? Now, the sort of unifying feature of lipids is that they do not mix with water, okay? They are what we refer to as hydrophobic. And I talked about this a little bit in a previous chapter. A molecule that's hydrophobic is by definition non-polar, which means that it has no positive or negative charges on it anywhere, and therefore there's nothing for either the positive or negative side of water to attract to. So hydrophobic molecules like fats and oils do not mix with water because they don't bond with water. All right, now the reason why they largely are non-polar is because they consist mostly of hydrocarbons, and hydrocarbons are either entire molecules or large portions of these lipid molecules that contain nothing but carbon and hydrogen. Typically with a hydrocarbon, what you have is a carbon chain. It can be a very long carbon chain with nothing but hydrogen attached to it, and so on down the line here. Hydrocarbons are nonpolar and don't mix with water, but they're also very energy rich. Lots of potential energy stored in these covalent bonds that you see here in this molecule. All right, we'll see more about those in a little bit. Now, one thing to start out with here with lipids is that the most biologically important lipids are the fats, which we've already kind of discussed, another group called phospholipids, and then a third group called steroids, and we're going to hit each one of those as we go throughout this portion of the chapter. All right, let's talk about fats first. So again, fats are not built on this monomer-polymer plan, okay? What they have, or their basic structure, consists of a portion of the molecule, called a glycerol. Glycerol is a 3-carbon alcohol, and it has a hydroxyl group attached to each of the 3 carbons. And then what you have are either 1, 2, or 3 fatty acid chains. These fatty acid chains attach to the glycerol at each of those 3 hydroxyl groups, and the fatty acid chain is the hydrocarbon portion of the molecule. That's the portion that makes the entire molecule hydrophobic, nonpolar. That's why it doesn't mix with water, but also the fatty acid chains contain the potential energy. Think of them as like the fuel tank for that molecule. Let's take a look. So here we have a typical fat molecule, okay? So first of all, what you see here is the glycerol portion. It's a 3-carbon alcohol, 1, 2, 3. Remember I said alcohols have OH groups, and here we have 1, 2, 3 of them, 1 on each of those carbons. And the thing about these OH groups is that they represent attachment points for a fatty acid chain, such as you see right there, okay? So this fatty acid chain will attach to that carbon via a reaction between the OH group and the OH group here. Technically, this is a carboxyl group, COOH. We talked about that in chapter 4. one functional group, second functional group, reactions happen between functional groups, and that's certainly the case here. But technically, it's just the OH group on the carboxyl that reacts with the hydrogen on the OH here. They come away again as a water, so this is a dehydration reaction, but you get a bond formed between that carbon and that oxygen right there. That bond is called an ester linkage, okay, an ester linkage. So again, this is different terminology than what we saw with carbohydrates. There, the bonds that held those monomers together were glycosidic linkages. You don't have those in lipids. Instead, what you have is a bond, a covalent bond holding the fatty acid chains to the glycerol, and it's called an ester linkage, all right? So we have one ester linkage bond formed there. We could have a second here and a third here, and so we could attach 1, 2, 3 fatty acid chains to that glycerol. When you have 3, the molecule you get is called triacylglycerol, or you probably know it better as a triglyceride. Same thing. It's one of the main things they check when you have your annual physical. They do your blood work, they check your fat levels in your blood, they're checking your level of triglycerides, okay? So you can see that with all these hydrocarbon portions here, all these covalent bonds in the hydrocarbon, that is a lot of potential energy storage. And so that's the fuel tank of this molecule, okay? Let's look at a video.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in one or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, so the video brought up a very important point, the distinction between saturated and unsaturated fats. And everybody's probably heard about saturated versus unsaturated in terms of diet, what kinds of fats are healthier, that kind of thing. Typically we think of unsaturated fats like that you find in plant oils, like olive oil, for example, to be healthier to cook with than saturated fats like what you find in butter and certainly lard and things like that. Let's talk about why it is that these molecules are referred to as saturated or unsaturated. Well, it has to do with the number of hydrogens in the carbon chain. So I'm just going to draw this down here at the bottom on this slide with the video. So let's imagine that you have a small carbon chain like this, okay? And notice we have all single bonds between those carbons. That means this carbon can have 3 hydrogens attached to it. This one, 2, this one, 2, and this one again, 3. So, because we have only single bonds between these carbons, every carbon, while being single bonded to another, can have the maximum number of hydrogens. This can have 1, 2, 3. That's the max it can have while having a bond to another carbon. This one that has 2 bonds to carbon can only have 2 hydrogens. Same is true here. This one bond to a carbon, can have 3 hydrogens. So each of those carbons then has the theoretical maximum number of hydrogens possible. Carbon forms 4 bonds, right? Okay, that is a saturated fatty acid chain. Now, what about this situation? Okay, well now we have a double bond between these 2 carbons. So while this carbon can have 3 hydrogens, and that one can have 3, this one here can only have 1, and this one only 1. Why? Because they have a double bond between them. So that carbon has 2 bonds there, a third one to there, it has 3 bonds to carbon, only 1 more bond possible, goes to a hydrogen. Same is true on this one. So these 2 carbons are not saturated. They don't have the maximum number of hydrogens they could have. if this were a single bond, you could put another hydrogen there and another one there. Those two carbons would then be saturated. But as you see, we have a double bond right here, so that makes this chain unsaturated. But while that one's saturated and that one's saturated, this is considered an unsaturated fat just because of the presence of one double bond. And also notice that double bond gives that carbon chain a bend or a kink. That's very important, as we'll see here in a little bit. Okay. All right. So
Butter.
Some examples. All right. So here we have an example of saturated fat. So this is a kind of fat. It's called stearic acid. There you see the name of it right there. That's found in butter. And you have your glycerol here. You have 1, 2, 3 ester linkages with 1, 2, 3 fatty acid chains. And they are all saturated. All right, what is this zigzag pattern about? That is the carbon chain. There's a carbon there, 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 and there. There's a carbon at all the points, and they actually do form sort of a zigzag, which you don't often see in images like this. But that carbon chain is straight because there are all single bonds between those carbons. Same there, same there. That's a saturated fat. And again, because those carbon chains are saturated and straight, these kinds of fats tend to be solid at room temperature, like butter. Now, over here, we have oleic acid. Here you see the name of it. Oleic acid is one of the primary fats in olive oil, for example. It has 2 saturated chains, but 1 unsaturated. Note the double bond. Gives it a bend, as you see right there. So that entire fat molecule is unsaturated, and at room temperature, unsaturated fats tend to be liquid. Okay. Now, the major function of fats is energy storage, especially in our cells. All right, humans and other mammals store their long-term food reserves in specialized cells called adipose cells. Adipose cells. Adipose means fat. So an adipose cell is a specialized cell in mammals. It's only found in mammals that does nothing but store fat. All right. Other animals, other vertebrates in particular, like birds, reptiles, fish, amphibians, they do have fat, but the fat is not stored in specialized cells. It's stored in structures called fat bodies. They're not cells, it's just collections of fat in the body cavity. But mammals store their fat in specialized cells called adipose cells. And so all of the fat in your body is stored in adipose tissue, collections of adipose cells. And adipose tissue has a number of important functions in addition to storing up energy for the long term. It also cushions vital organs. So for example, your kidneys, which if you feel your lower back, the kidneys are located below your rib cage. They have no protection from bone. That's why if you get hit there, it hurts so much. But they do have some padding in the sense that they're covered in a layer of fat, a fairly thick layer of fat or adipose tissue. So that's an example of adipose tissue cushioning important body organs, for example. Another important function of adipose tissue in our bodies is that fat retains heat. It's very good insulation. It helps our body to retain heat. So it's this adipose tissue that we have, in particular under our skin, on top of our muscle, but also in certain other parts of the body, is very good insulation against the cold. Okay. All right, but in terms of biochemistry, now we're talking about adipose tissue or fat tissue as being a long-term energy storage molecule. And the reason why it's a long-term energy storage molecule, fat is because it stores up so much more potential energy than proteins or carbohydrates do. A gram of fat stores twice as much potential energy as a gram of protein or as a gram of sugar. So if you're consuming more calories on a daily basis than you need, your body doesn't get rid of that excess calories. Okay, what it does is it stores it eventually for the long term as fat. It's more efficient to store its fat because a smaller volume of fat stores more energy than a larger volume of sugar or protein. It's a more efficient way to store it. This is also why if you're trying to exercise to lose weight, you have to do so much exercise to lose weight, because a small amount of fat can power your body through an hour-long workout on a treadmill or an elliptical or a run. And so you don't lose a lot of weight doing that. It takes a lot of work to get rid of that fat because it's so energy rich. Okay. Now, next thing we're going to talk about is a special kind of fat molecule called a phospholipid. Okay, phospholipid. Phospholipids have structural roles to play in our bodies. They're not there for energy storage, okay? Triglycerides are there for energy storage. Phospholipids are not. Phospholipids are the primary building block or building component of our cell membranes. So they're critically important because without phospholipids, we can't imagine how you could have cells, because you couldn't build cell membranes, which is the outer margin of a cell that separates the internal environment from the external environment. Now, what you typically have with a phospholipid, and this is the way they're kind of drawn in textbooks, is that you have what's called a head on the molecule, and then you have two fatty acid chains, okay? The head is where the glycerol portion of the molecule is contained. We're talking about a lipid here, right? So it has a glycerol, but then you have two, always two, and only two fatty acid chains connected to that. Now, the thing about the head, the phospholipid head as it's called, is that it's hydrophilic. So it's polar. That means it attracts to water. The tails, as we've already seen, are hydrophobic, non-polar. They do not attract to water. So here we have a unique feature of phospholipids, something that other fat molecules simply don't have. Part of the phospholipid molecule is polar, part is nonpolar. Part of it attracts to water, part of it does not. So in a single molecule then, you have part of it that's polar and therefore attracts to water, part of it nonpolar and does not. That condition, part of the molecule liking water, part of it not, is referred to as amphipathic. So phospholipids are amphipathic molecules. Part of them likes water, part of them doesn't, and this is key to phospholipids being able to be used by our bodies to build cell membranes. Let's take a look. So what you see up here, first of all, top right, is kind of what I drew in the previous slide, right? You have the hydrophilic head, the part that likes water, hydrophilic, water-loving, and two fatty acid chains that are hydrophobic, okay? Now, if we look at this, you can see a space-filling model. This is the hydrophilic head. There's your glycerol, but notice it also has a phosphate group and something called a choline. Don't worry about those in particular, but these are the portions that give that hydrophilic head its polar nature, all right? They're polar. Then you have two fatty acid chains, and notice that this one on the left is saturated, it's straight, and this one on the right is unsaturated, has a bend or a kink in it, okay? Now, on the far left, we have a structural formula. So here again, you see the two fatty acid chains, one saturated, one not. You can see the double bond there making this one unsaturated, and then you have the hydrophilic head. There's your glycerol, a little modified, phosphate with a negative charge, choline with a positive. These two additions to that glycerol are why that head is hydrophilic. So here's the thing. If you have pure liquid phospholipid and you put a drop of it in water, the heads will attract to water because they're polar. The tails will shy away from water in the same way that oil and water don't mix. And so what you get is an automatic structure formed. It's called a phospholipid bilayer. You see how you have one layer of phospholipid here and a second layer there, and their tails are backed up toward one another, with the heads coming into contact with water on both sides. That is the basic structure of our cell membranes. Our cell membranes are phospholipid bilayers, and the reason why that bilayer forms is because the phospholipid is amphipathic, part polar, part nonpolar. And so the tails tuck inward, the heads point outward to water, and you get a bilayer structure just as a result of the way that the chemistry works. And it turns out this is the basic structural framework of all cell membranes that we see on life in life on Earth. So now you can see that if phospholipids weren't amphipathic, if they didn't have a polar portion, they would not form that bilayer structure, and we couldn't have cell membranes as we know them. Okay. All right, so the existence of cells then depends on phospholipids. Very important. Okay, next category of lipids are steroids. Now, everyone's heard of steroids. We tend to think of steroids in terms of like athletic doping scandals, athletes taking steroids to improve their performance artificially, that kind of thing. Those are artificial steroids, but there are natural steroids found in our bodies that play very important roles. One great example that you'll all be aware of is cholesterol. Cholesterol is a fat molecule. In particular, it's a steroid. Cholesterol plays an important structural role in our cell membranes. It has other roles to play as well, but it plays a very important structural role in our cell membranes by helping the cell membranes to hold together. Okay. Now we know this is the context in which we hear of cholesterol. If you consume too much cholesterol in your diet, it can build up in your blood, may contribute to cardiovascular disease, that kind of thing. And there are actually two types of it. There's what's called LDL cholesterol and HDL. The LDL is the bad one. This is the one that you don't want to have a lot of. I tend to think of or remember it as starts with L, lethal, right? A little overly dramatic, but it's not good for you. HDL, think of it as healthy, right? These actually stand for low density lipoprotein and high density lipoprotein. Don't worry about that, but that's what the L and the H stand for, low and high. The HDL is what you want to have more of. And they always look at your ratio of those two when they do your blood work for your annual physical if you have that done. But cholesterol does play an important role in our cell membranes. Okay, so what is the structure of it? This is the structure of cholesterol. And what you see is that right off the bat, it looks very different than the other fat molecules we've been talking about. First of all, there's no glycerol portion there. What you have are 4 fused carbon rings. Then you have a fatty acid chain coming off of that with some methyl groups here and so on. If you recall, I showed you something very similar to this back in, I think, chapter 2. I showed you the structure of estrogen and testosterone. They also had 4 fused carbon rings. And the reason why there's so much similarity between estrogen, testosterone, and cholesterol is because cholesterol is the molecule that is used to build estrogen and testosterone. It's a precursor to those two sex hormones, but it is a fat molecule, okay? All right, just a very unique fat molecule. Okay, so the next, that sort of wraps it up as far as the fats go. We're going to see more about cholesterol in, I think, chapter 7. We talk about, or maybe chapter 6, we talk about cell membranes a little bit. We'll see it again there. But for now, we're going to end on lipids and move into proteins. Now, I may have to stop partway through proteins and then do the rest as a third video lecture, but that's okay. All right, so proteins are next, and there is simply no way to understate the importance of proteins. Our biochemistry, as we know it, simply could not exist. It could not work the way that it does if we didn't have proteins. They didn't exist, all right? Now, proteins are super abundant in our cells. They account for more than 50% of the dry weight of most cells. So you take some tissue, organic tissue, and you dry it out completely. Usually 50% or more of the weight of that tissue is due to protein alone. And it's even higher when you look at specialized tissues like muscle tissue, for example, which has a lot of protein in it. Now, proteins have a number of different key sort of functions or roles to play in our biochemistry. One of them is that they behave as enzymes. And we talked about this already in the first video lecture for this chapter. Enzymes are specialized proteins, okay? They are proteins that speed up chemical reactions. That's one key function of proteins. But then we have another of other important functions in proteins as well. Some of them include defensive functions in our bodies. They play an important role in our immune response, for example, against invading pathogens, viruses, bacteria, that kind of thing. Some proteins have roles in terms of storage. They store things. We'll see more about that. Some proteins play transport roles. Some play an important role in communication, allowing cells to communicate with each other. Some play an important role in movement, okay? And then others play a very important sort of structural role. They help to build parts of our bodies. And we're going to see examples of all these starting here next. So let's take a look. All right, so first of all, we have enzymatic proteins. So proteins that act as enzymes, all right? So basically the way this works is that really, no matter what kind of enzyme you're talking about, each enzyme has its own characteristic three-dimensional shape. They're all unique in that way. We'll talk a little bit later on why that is. But the enzyme, which you see here is a sort of purple pill-looking structure, the enzyme has a site on it called an active site. Usually it's a series of depressions or a depression or a trench or a trough or something like that. And there's another molecule that fits precisely into that active site. It's almost like this molecule is the key and the enzyme is the lock. They fit together. Or like a hand going into a glove, for example, or puzzle pieces, if you want to think of it that way. And so what happens is that if you have, let's say, a disaccharide, which is what this green molecule is, there's a glycosidic linkage there. That disaccharide fits into the active site, and the enzyme catalyzes the hydrolysis reaction that breaks that bond. We talked about hydrolysis reactions in part 1 of this video lecture, right? Using a water molecule to break a bond. Well, I said then an enzyme helps this, and now you actually see the enzyme there. Okay, we'll see more about that in a little bit, how they actually work. We have storage proteins, okay? So these are to store amino acids. So I failed to mention, we got back into proteins here a moment ago, that we're now back into talking about macromolecules that are built on that monomer-polymer plan like we talked about with sugars. So you take a series of monomers, link them together, you get polymer. In the case of proteins, the monomers are called amino acids. Those are the monomers, all right? Okay, we'll talk about the polymers more in a minute, but basically we have proteins that act as nothing more than a storage tank or a reservoir for amino acids. So think about milk. We know that milk is a good source of protein, right? You drink it for protein, that kind of thing. One of the primary storage proteins in milk that stores amino acids is casein, right there. That's one of the primary protein or amino acid storage proteins in milk, okay? So you drink cow milk. You're not drinking cow milk because you need cow casein to build your muscles, for example. You need the casein in that milk because your body needs amino acids. Your cells, when you drink milk, will take that casein in. Hydrolysis reactions will separate all those amino acids from each other that are used to build that casein, and then your bodies rebuild those amino acids into human proteins. The same thing is true with the ovalbumin that you get from eggs, for example, or the chick developing in the egg. The ovalbumin, which is the white part of the egg, all right, the white of the egg, is a storage of amino acids for the embryo. The embryo takes the ovalbumin in, breaks the amino acids apart, builds them into the various different kinds of proteins it needs so it can grow and develop. All right. Defensive proteins, upper right here. So these are proteins that are primarily involved in our immune response, our immune system. Okay, so we have proteins called antibodies, for example. And this is timely given, the COVID that's going around right now with the pandemic. Antibodies are signaling molecules. So antibodies are proteins that are circulating around through your bloodstream, for example. Let's say a virus or a bacterium enters your body, enters your bloodstream. Those antibodies will attach to the virus or the bacterium, as you see right here, right? And in sort of military parlance, they light it up. What they do is they signal to your immune system, macrophages, white blood cells, that sort of thing, that this virus is foreign. With the antibodies attached to the virus or the bacterium, your macrophages, for example, can see that virus and therefore attack it and hopefully destroy it. So that's just one example of how proteins are important in our immune system. Defensive proteins, they're antibodies. Okay, they can inactivate and help destroy viruses and proteins in that way. Here we have transport proteins. So these are primarily for transporting substances, primarily across cell membranes. So here you have a cell membrane. Remember that phospholipid bilayer I talked about. You'll have lots of proteins embedded in these membranes, and they allow for solute molecules to pass through. Remember, In that phospholipid bilayer, you have those fatty acid chains that back up to each other. And the fatty acid portion of that lipid is hydrophobic, nonpolar. So you end up with a fairly thick hydrophobic barrier in the middle of that membrane across which solutes that are dissolved in water can't move. If they're dissolved in water, they're by definition polar. Polar and nonpolar don't mix. They separate like oil and water. So if you have a solute outside of a cell that needs to get in and it's dissolved in water, it's by definition polar, hydrophilic. It can't get through that barrier, so it'll go through a transport protein. Think of the transport protein as sort of providing a route of safe passage to it, okay? And we're going to see lots of examples of those in the upcoming chapters. All right. Next, we have hormonal proteins. So hormonal proteins are proteins that act as hormones. They are messenger molecules, okay? They help to coordinate an organism's activities, cellular activities. So think of a hormone as a protein that's released by a certain gland in your body, let's say. And that protein will have a target tissue somewhere in the body where it needs to send its message. So the protein will bind to the surface of those cells in that tissue, and through a very complex chemical reaction, that message gets passed into that cell. All right. Now it's complex, and we won't get into a lot of details of it. But just think of the hormone as a protein that's sending a message, right? Here's a good example. Insulin. Insulin is secreted by the pancreas, and it's a signaling molecule that tells your cells to begin to take up glucose. So glucose is a sugar that's found in our food, and within minutes of food entering your stomach, your pancreas begins to crank out insulin. Insulin floods into your bloodstream. It begins to it's a messenger that begins to tell your cells to get ready to take up the glucose that's getting ready to flood into your bloodstream through the digestive process. So it primes your cells to take up insulin. Okay. People who have diabetes don't make insulin properly or not at all, and therefore their blood sugar levels can go up dangerously so when they eat food because the blood sugar doesn't go into the cells. They can take insulin in an injection or whatever, and in that way their cells can take up that sugar. But they have to monitor their insulin levels, obviously, and their blood sugar levels. Okay, receptor proteins over here. So receptor proteins, so you have these little purple proteins here embedded, say, in the surface of a muscle cell, right? We have a nerve cell here. A nerve cell will have a junction with a muscle cell with a microscopic gap between it. It's called a synapse. Not really important for our purposes now, but there's a gap there. Signaling molecules will be released from the end of the nerve cell when a nerve impulse moves down that nerve cell, and the signaling molecules will diffuse across that space and attach or bind with receptor proteins, which then sets up that nerve impulse inside the muscle cell, causing that cell to contract, let's say. So receptor proteins are really important, especially in our nervous system, for allowing cells to respond to chemical stimuli, okay? Here we have contractile and motor proteins. So think about your muscle cells, right? Let me zoom in a little bit on this. So muscle cells have a functional unit inside them that allows the entire cell to contract, okay? These vertical bands that you see here are those functional units. They're made of two kinds of protein, actin and myosin. The myosin forms a sort of central cylinder around which you have actin fibers. So these little heads here, called the myosin heads, will attach to these actin fibers, and they will drag the actin fibers during a muscle contraction this way and this way, causing the muscle cell to shorten. It's a chemical reaction then between these two actin and myosin proteins. That's what a muscle contraction is. When you raise your arm, your bicep contracts. It's a chemical contraction at the cellular, or chemical reaction rather, at the cellular level that causes the cell to contract. If you take a physiology class, you'll talk about muscle physiology. You'll learn how it all works. It's very complex, but basically, contractile motor proteins allow for movement. In this case, muscle contraction. There are also other proteins that essentially help to move things along little monorail tracks or train tracks throughout the cell. They move them from one end of the cell to the next, to the other, for example. We'll see examples of that in the next chapter. Okay, but those are proteins that are called motor proteins that move things around inside cells. And finally, we have structural proteins. Structural in the sense that they help to build things or provide support. Keratin is a good example. Keratin is one of the primary structural proteins that you find in invertebrate animals with any sort of extensions off of the skin or from the skin, like hair, feathers, hooves, horns, antlers, claws, nails, all those sorts of things are really extensions of the skin, but they're all made primarily of keratin, which is a very strong, long-lasting structural protein. Okay, insects and spiders use silk fibers to make their cocoons and webs. Right, that is there are certain kinds of structural proteins in that make it very strong. Another example, collagen that you see here, is sort of a rope-like protein that you find making up connective tissue, in particular in our skin. Skin is very, very tough, very resistant to tearing and that sort of thing, largely as a result of its high collagen content. So these are just some examples of structural proteins. There are lots of others as well. Okay. All right. So last thing to talk about, and I kind of already hit on it before already, is the enzymes. So of all these different categories of of proteins we've talked about here, while they're all important, biochemically the enzymes are the most important, because without them you couldn't have biochemistry, and without biochemistry you couldn't have life. Enzymes are very important molecules that are referred to as catalysts. Okay, catalysts speed up a process. You can think of gravity as a catalyst. If you're perched precariously on a skateboard on the peak of a hill, right? Gravity could speed you down one side or the other. It's a catalyst in that sense. Proteins are biochemical catalysts that speed up chemical reactions. Most of the chemical reactions in our bodies don't want to run very fast at a temperature of 98.6 Fahrenheit and a pH of 7.2 or 7.3. Those are fairly benign conditions. And if you think about it, in a chemistry lab, if you want to speed up a reaction, one of the best ways to do it is to heat it or to change its pH by adding acid or base. Well, our cells cannot tolerate changes in temperature like that or changes in pH, so it can't do that. So how do our cells speed up chemical reactions? Through catalysts, enzymatic proteins that speed up biochemistry. Okay? All right. Well, I think that's a good place to stop for this video. The third video lecture should be the last one, I hope, for this chapter. We'll start to talk about protein structure. Okay, and again, these are monomer-polymer plan. Again, we'll talk exactly how it is that the monomers of proteins are used to build large, complex protein molecules.